Well, I guess I take this opportunity to welcome you on behalf of the Cancer Center, and we're really glad you're here, and we're really excited about this effort, and excited about the afternoon and tomorrow. And what Raul did was to say, who in this community knows the least amount about mathematical data analysis? And he said, Corey. And so he asked me to chair the session. I'm just kidding. We collaborate a lot with these guys. So without further ado, I'm going to start the afternoon session, and it's my pleasure to introduce Olivier, who's come across town from Weill Cornell. And for those of you who don't know, Olivier Elemento, he's a leading computational biologist and one of the real leaders at Weill Cornell Medical College in the area of cancer systems biology and systems biology and genomics. So welcome, Olivier.
Thank you. All right, I'm going to try to find my slides now. It's a Windows computer, so good luck to me. Oh, here you go. All right. So first of all, thank you so much for the invitation to be here to present today. I wanted to describe activities that are happening within the Englander Institute for Precision Medicine at Cornell, which I have a privilege to direct. And it's an institute that focused on essentially establishing the precision medicine paradigm, starting with cancer, but now really branching out, using the same paradigm outside of cancer, for example, in cardiovascular disease and other diseases. So the talk today is going to have multiple components. I'm going to try to show you different aspects of the program that we're running, focusing on profiling of clinical samples, focusing on analytics, which is required to integrate the information from the clinical samples to make predictions about how to treat patients, and then modeling, which essentially is an orthogonal sort of way to explore the precision medicine paradigm, not only looking at genomic information, but trying to essentially use models to get a functional assessment of what's happening within tumors, and use this again to direct cancer therapy. A lot of what I'm going to talk about today is really sort of meant at Cornell to be used for clinical care, but in the process of sequencing lots of genomes, as you'll see playing with these models, we're generating a tremendous amount of information and data that's fueling a very large number of research programs across the school, across the college. And I'll try to highlight some of these programs as well moving forward. So I'm going to give you a brief overview of the talk. So I'm going to tell you about the Cancer Precision Medicine program at Cornell. I'm going to highlight what I think we've learned in the past few years, sequencing of these genomes, and using these concepts of tumor organoids that I'm going to present. But I would really like to also highlight challenges and to give you a sense of how we are trying to address those challenges in the field. Challenges, for example, consist of how do we identify what treatments are most effective for individual patients. You'll see we're using genomics to get to that problem, but also the concept of patient-specific avatars, which is basically trying to get, essentially trying to make copies of somebody's tumor in a dish, in vitro, and doing high-throughput screening on these copies, and essentially identifying what patients are likely to respond to in terms of drugs or drug combinations. But I also would like to highlight work that we're doing in terms of rationally identifying drug combinations, trying to understand mechanisms of response to drug combinations, modeling what's happening within tumors so that we can, even without having to do any testing, predict what drug combinations patients are going to be sensitive to. And obviously, as we move forward, as we have the ability to really dig very deep into what's happening within cells, within also tumors, looking at tumors at a single cell level, there's a major need to integrate multiple data sets, multiple data types, to be able to make very predictive models about what patients are going to respond to. And I'll try to highlight also some of these components within my talk. And the talk is really going to continuously combine analytics and experimentation. That's going to be a theme of the talk, the need to combine those two components across the board. So this is an overview here of the precision medicine program at Cornell. We've been studying the program with advanced cancer patients, patients who are running out of options at the clinical level, being essentially resistant to all standard therapies, and where it's very compelling to attempt to use precision medicine, genomic sequencing, and other opportunities to identify what to do next at the clinical level. So in those patients, what we do is we get new biopsies. So we're getting essentially a new source of tissue within those patients. The goal here is really to try to understand what's driving the cancer at this moment. We're not interested or less interested in what used to be driving the cancer several years ago, potentially before the tumor came back as resistant and metastatic. We want to know what the tumor looks like at the time that the patients present to the clinic. So we're doing new biopsies from patients. Every time we get tissue from a patient, we also get blood. And when we sequence, as I'll show you, tumors, we also sequence control DNA from blood. And the idea, obviously, for us is to look at mutations that are specific to the tumor, highlight mutations that are likely driving the cancer, but also more and more understand what kind of mutations are inherited from the parents of these patients, what kind of mutations are potentially predisposing for cancer, or potentially even helping fuel cancer and fuel the aggressive nature of cancer in those patients. And as you'll see, we have some results also in that space. So once we have tissue, we do multi-omic profiling. So we do whole exome sequencing as a default test for all patients at Cornell. The whole exome sequencing is looking at 22,000 genes. Again, tumor and normal sequencing. Whenever we can, and we do this now for a bit more than 50% of patients, we do RNA-seq. We also look at what genes are expressed within the tumors. As you'll see, this gives you very valuable information when it comes to understanding what pathways are active within tumors, but also more and more, it gives you information about the microenvironment. By looking at genes that are specific to, for example, specific immune cell population, we can deconvolute the microenvironment of tumors and, for example, identify the fraction of T cells that you have within the tumor and what kind of T cells you have within the tumor, what kind of macrophages you have. And this information is also very valuable when it comes to identifying treatment for patients. For example, identifying which patients are likely to respond to immune therapies. The data that comes out of this profiling effort goes into a report. I'll show you examples of the report. What goes into the report is also digested, explained, analyzed in sessions that we call tumor boards, where we have multiple experts from different fields come in the same room and look at the same genomic information and try to identify, based on all of the comprehensive characterization, what is the best option for each patient, what's the best clinical option. And based on those reports, the clinical recommendations are made and patients are treated accordingly in most of the cases. So I'll go over some more details about the program in the next few slides. The key component of this program is a New York State approved, CLIA approved whole exome sequencing assay. We've been one of the first centers in New York to get approval for whole exome sequencing in the clinic. This was quite a bit of work, quite an endeavor, sequencing essentially hundreds of samples, establish credential the assay, make sure that the assay is reproducible, reliable, and so on. We've got to that point and now can really offer whole exome sequencing in the clinic, and for us it's a major achievement. How we report results to patients is obviously very important. We report results to patients but also to physicians. We create these very personalized reports that you see here with multiple types of information. We have pathology information on those reports, and we have mutational information, somatic mutations broken down in different categories of actionability. So the highly actionable mutations are at the top of a report, and then you have the least actionable mutations at the bottom. In the middle, you have what we call driver mutations, mutations that are likely to be important for the cancer, likely driving the cancer, but not known to be targetable with drugs at the current stage. And so we've produced now about 1,500 reports like this. So every time we sequence a cancer, every time we sequence a tumor, we produce a report of this kind, and the report goes to the physician, to the patients. and also goes into the EHR as structured data enabling queries in clinical trial population moving forward. It's just sort of multi-page report here. The way that we populate the report is essentially when we sequence somebody's genome, we get a list of mutations, and we look up these mutations into a precision medicine knowledge base. It's essentially a database of cancer mutations focused on clinically actionable mutations that we populated over the years, and that has a lot of information about thousands of mutations. And essentially at this stage, for the report itself, we essentially just look up mutations in this database and populate the report accordingly. Obviously, the interpretations of the mutations can be changed. The pathologist who signs out the reports can potentially change the interpretations by looking at, for example, combinations of mutations and their significance. So as I was telling you, we've been doing this now for several years. We've sequenced a bit more than 1,500 patients. As you can see in this picture here, the patients are really all over sort of the map in terms of types of cancers. We're not focusing on one type of cancer. We're sequencing every tumor type. These are all metastatic cancer patients, but really they are really representing every single cancer type. And we see accordingly a very dramatic amount of diversity when it comes to the tumor types. And again, keep in mind that these are metastatic cancer patients whose tumor has evolved in addition to being diverse, in terms of organ or origin in the first place. So what have we learned? So I'm just going to show you a couple of vignettes about things that we think that we learned that are important. We've been able to sequence, in many cases, more than one tumor per patient. In some cases, when we look at rapid autopsy cases where essentially some patients consent to giving us access to tissue after they pass away, we can actually get access to up to 10, 15 tumors from the same patient, all metastatic different sites. And that allows us to reconstruct essentially the evolution of tumors. Allows us to understand what's common between multiple tumors in the same patients, the mutations that are common, the mutations that are specific to individual tumors. And we see a tremendous amount of evolution within the same patients. We see a lot of branched evolution, where, for example, the first tumor that you see in a patient is dramatically different from the relapsed tumor that you see when the tumor comes back. Very few shared mutation. Some enough shared mutation that that the two tumors have a connection. They've come from the same sort of origin, but they've evolved essentially across different branches. And we see this now across multiple tumor types. This is an example of bladder cancer that we published a couple of years ago now. Another thing that we've seen, not looking at somatic mutations, but looking at the germline, looking at mutations that are inherited in those patients, we see a tremendous number of patients, of metastatic cancer patients with DNA repair mutations in the germline. In breast cancer, for example, as you see here, it's about 25% of patients that have essentially a DNA repair mutation in the germline. And these numbers are really high, much higher than we expected. And again, several studies are showing the same patterns. But really, I mean, it's the preponderance of these mutations in the germline that are likely to be either sort of predisposing to cancer or potentially fueling the aggressive nature of the cancer. Remember that these are all metastatic cancer patients. Is much higher than expected across the board. And some of these mutations are interesting not only from sort of a family, testing point of view, origin of a tumor, but obviously because there are also therapeutic opportunities. It's more and more common that some of these DNA repair mutations give rise to weaknesses within the tumors, using, for example, PARP inhibitors that have a potential to selectively kill tumors that have this kind of germline mutations that's potentially driving them. So again, opportunities in addition to sort of interesting findings across the board. Since we do, in addition to whole exome sequencing, we also do RNA-seq. We also sequence the genes, essentially quantify the genes that are expressed within tumors. We can essentially correlate mutational presence and, for example, activation of specific pathways. This turns out to be very important, especially in metastatic tumors, because we see a lot of, as I said, tumor evolution. So we see a lot of tumors essentially colonizing, different sites, different locations. And we see a lot of tumors that seem to have driver mutations, mutations that you would think of as potential driver mutations. But when you look at the genes that are known to be downstream of those mutations, those genes are not expressed at all whatsoever. So basically, it tells you that very often you have driver mutations that are likely to be inherited from a previous stage where the mutation was really driving the cancer. But when the tumors move to a different location, those mutations stop being driver mutations. But they are still there, inherited from the past within the tumors. And so it's very important to have a kind of a functional readout of the mutations so that we can tell whether the mutations can be targeted or not. And that's something that we're learning actively in this program. This is just here one case in which we found an ERB2 gene fusion actually in this patient. And it was reassuring that when we looked at genes that we know are downstream of ERB2, we saw that those genes were actually highly expressed within those tumors and more expressed than in control samples. So it basically tells you that ERB2 in this particular patient is really active because you see that downstream you essentially have activation of a pathway. So that's essentially two levels of evidence that really obviously converge into the same concept, and obviously give you a lot of confidence that ERB2 is a targetable mutation in this one patient. And again, this may be different in another patient. Just very briefly, obviously, because one of the goals of the program is a clinical goal, we're looking at outcomes, we're looking at situations where the whole exome sequencing was helpful, and we have many such cases. This is just one case here, which I'm going to present very briefly. It's a metastatic bladder cancer patient that was not responding to therapies anymore, but in which, using whole exome sequencing, we were able to find a HER2 amplification, which in bladder cancer a few years ago had not really been described, in a very sort of frequent way. But obviously, HER2 amplification is very frequent in breast cancer. In breast cancer, HER2 amplifications are targetable using Herceptin. So what the clinicians decided to do in this particular case is to give Herceptin to this patient. And Herceptin, as you can see here, essentially had a dramatic response. The tumor melted away, and this patient is still alive several years after start of treatment. She continues to be on Herceptin, but at least 2 years after the start of treatment, she's still absolutely healthy. I met her recently actually, and she's very very healthy and doing really well. So just an example of an unexpected discovery of a mutation that's targetable that gives rise to really a complete response, and that highlight the potential of this whole precision medicine paradigm. And as I say, we have several examples of such success now. That being said, if you look across the board, it's probably only about 15-20% of patients for which we find a targetable, actionable mutation. That's the reality of genome sequencing. It's only a minority of patients who can really benefit from the sequencing itself. So obviously, we're continuously looking for opportunities to expand the number of mutations, number of cases where we can do something in those patients. And that's where a lot of challenges occur, and we're trying actively to address those challenges. So as I mentioned at the beginning, challenges include, for example, how do we identify what treatments are most effective in patients? How can we predict which treatment is going to be most effective in individual patients? How can we identify combination therapies, right? There's no doubt that the future of cancer therapy is through combination therapy. But as we essentially start having a situation where you have lots and lots of molecules to choose from, even within individual types of tumors, deciding how to combine those therapies, including deciding how you combine them in a personalized way, is very difficult. It's just lots of combinations to test, potentially. So we're trying to think a little bit, ahead of the game, trying to think about how we can predict how to combine molecules and drugs in individual patients. I'll show you a couple of results about this. And obviously, we would like to improve our understanding of disease biology in general and improve actionability, and I'll show you more also slides about this. So one of the main sort of components of the program, which is also something that distinguishes us from what others are doing in the field, is that we've been able to optimize technologies to take tumor cells from patients, fresh cells, and essentially grow them in a dish. The goal here is really to essentially make mini tumors from the same patient, make as many mini tumors, essentially copies of a tumor from the original patients, so that we can do essentially high throughput drug testing on those mini tumors, and essentially try to see what's effective at killing cancer cells, irrespective of the genomics, just essentially a functional sort of screening of what's effective at killing cancer cells. And again, not only looking at single agents, but also potentially drug combinations. So this is the branch that's at the bottom. We really try to combine genome sequencing, whole exome sequencing at the top, with this tumor organoid program, which we also combine in as many cases as possible with PDX derivation so that we can test results from the organoids also in vivo. Obviously, the timeline, the kinetics, the dynamics of PDX models is a bit longer and not always compatible with sort of clinical application, which is why we really would like to push the concept of tumor organoids as far as we can. But initially, to validate the concept, we have to use PDX models, at least as in the first steps, to be able to test and validate the concept. So these are examples of organoids. So again, what you see on the right side here are mini tumors from one patient. Essentially, these are copies of the same tumor from one patient. We know that they are good copies because we sequence the genomes of these copies. We do whole exome sequencing on them. I'm not going to show data here, but we have very good data showing that because of the way that because how we grow these cells, essentially in 3 dimensions, we're not imposing too much pressure for these cells to change and to evolve and to become different. They don't have to adapt too much. And as a result, they don't change their genome dramatically. And so these copies that you see on the right side are actually pretty faithful copies of the original sort of tumor that you see in the patient. The strength of having multiple copies like this is that you can essentially test one different drug in each of the copies and see what drugs or drug combinations are effective at killing each one of a copy. And we do this using robotics. This is here just some data showing the success rate. Obviously, one of the caveats of these programs is that we're not always successful. It's not in every patient that you can derive an organoid growing in a dish, and even less so a PDX model. In some tumor types, we actually have very low success. But in other tumor types, and this slide here is a little older, like for example, in colorectal cancer, we are about 80% of the time successful in deriving an organoid for each individual patient. So in some tumor types, the paradigm is working really well. In other tumor types, we're still refining the paradigm, trying to improve how we grow these tissue in vitro. But we're making a lot of progress, and I can speak to this at some point. The strength of these organoids is that once we have them, you can bank them, you can essentially freeze them, and you can use them for other things. So we use them primarily for personalized medicine, precision medicine, but you can also use them for drug screens, right? Let's say, for example, that you have a molecule and that you want to know which tumor types, which cancer the molecule is most effective at killing. You can actually take all these organoids from different tumor types from the fridge, grow them again, and then treat each one of them with the same molecule. And basically find out, for example, that the molecule is effective in prostate cancer, but is doing nothing in breast cancer, just as an example. So it also gives you this ability to screen molecules, and obviously it's very useful and effective in many ways. And the organoid number that we have now in the biobank is about 50, somewhere between 50 and 60, and obviously it's growing every month significantly. So I was telling you we're doing high-throughput screening of single agents. We've been doing this in the past few years with a company in Seattle, but we're now in the process of bringing the technology, the robotics, to do the screening in-house. We would like to be able to have the flexibility to do the screening in-house so that we can try many more drugs, many more combinations, co-cultures of different kinds, and that's why we bring the technology in-house. But this is results from screening of about 120 drugs, FDA-approved drugs, using this company that we've been using in Seattle. So these are what you see here is 2 different patients. This is single-agent screens. 2 different patients. They share something. They both have colorectal cancer. You see the mutations that they have. They're not targetable mutations, so there's no kinase or HER2 or so on. These mutations are not thought to be targetable. But what you see very clearly in those high-throughput screens, and then what you have to look here is really the negative Z-scores here. So the scoring of individual drugs, obviously has multiple components. The point here is that the more negative the Z-score, the more effective the molecule is at killing these tumor cells in vitro. So what you're seeing in the patients at the top here is that the most effective drug in this particular patient was trametinib. It's a MEK inhibitor that's obviously highly effective in this one patient here. What's really interesting is that in this one patient here, this same drug is actually not effective whatsoever. Right? It's basically in the middle, sort of thing. But you have in that patient actually other drugs that are quite effective in this patient. So 2 different patients with the same disease as you can see, have a very dramatically different drug response profile across those compounds. What's interesting is you can combine, so you can use the same high throughput screening as a way to look for drug combinations. So what we do is we take the top hits from the first screen, and then we combine those top hits with the same 120 drugs, so as to identify the most effective drug combinations involving the initial highly effective drug in the first place. So in this one patient here, for example, you see that trametinib in combination with celecoxib was highly effective at killing those cancer cells. This is trametinib alone, as we discussed, it's effective. Celecoxib, on the other hand, is actually not effective at all whatsoever. But it's only in combination with trametinib that suddenly we get synergy and you get high effectiveness in terms of killing cancer cells. And again, we're getting, likewise, a very different sort of combination also in this particular patient here. So these high throughput screens, which we've done now for tens of patients, are revealing a broad amount of diversity in terms of response to molecules, and really sort of highlight the fact that you need to combine those screens with genomics to be able to really understand the different ways by which these tumors are going to respond to therapies. The good thing is that in some cases you get good agreement. So this is a glioblastoma patient here that had a PTEN loss and a PIK3CA mutation amplification. And according to the high throughput screen, indeed responded really well to mTOR inhibitors and PI3K inhibitors. So this is the situation here where obviously you have high confidence that these molecules have a good chance of working in patients, if you were to treat them. We are not yet at that stage. We have not yet used this paradigm here to treat patients. We are working on it. The first step is to essentially do the high throughput screening in a clear environment. And this is, what I was telling you. The robotics that we're bringing to Cornell, we are essentially building a high throughput screening core that's going to be clear, certified from the start, so that we can essentially use this paradigm as a way to treat patients moving forward. We are really trying to innovate when it comes to building organoids. There's great potential to build better organoids. For example, we're trying very hard to build organoids with a vasculature by essentially co-culturing tumor organoids with endothelial cells. We have great results. In fact, a paper in Review Nature about this showing that the organoids, when they are co-cultured with endothelial cells, the endothelial cells that you see in red here actually self-organize into blood vessel-like structures. They're not functional blood vessels, but they self-organize in this way. And what's really sort of striking is that the tumor cells that you see in green here also self-organize. We essentially try to get as close as possible to the vasculature, essentially sort of trying to get advantage of the survival signals that the vasculature is sending to them. And again, in data that I'm not showing here, this time in T-cell lymphomas, these are colorectal cancer organoids here. But in T-cell lymphomas, we're also doing high-throughput screens of tumor cells with and without endothelial cells, and we're getting dramatically different drug response profile with co-culture and without co-culture with endothelial cells, indicating that those survival signals that these endothelial cells are sending to the tumors are also very important when it comes to predicting, understanding what tumor cells are going to respond to in terms of your molecules and drugs. So this is, really a whole new area of research that is going to be very important, how to build better organoids. Another thing we're doing is also to build immunocompetent organoids. We're trying to seed organoids with immune cells, T cells, and a variety of immune cells so that we can essentially try immune therapies on organoids. And that likewise is really the future of organoid utilization, especially as a clinical tool. This is also just one slide of another innovation where we started growing tumor organoids in zebrafish. It turns out that zebrafish embryos don't have an immune system yet. They're developing it a little later during development. But they have a lot of survival signals that human tumor cells actually like quite a bit. That allows us to essentially grow human tumors within zebrafish embryos. And because the fish is transparent and fish has vasculature that you can see actually in the tumors, these are a great system for high throughput screening, but also to study the interplay between the vasculature and the tumor cells. So a great tool for research, but also potentially, who knows, for clinical applications at some point. Just quickly another vignette. So this is trying to sort of leverage now mathematics and modeling a little bit more. we've been trying to do some work to predict how to combine molecules. So essentially, you can do high throughput screening. You can essentially do a lot of experiments that indicate what combinations of drugs to use. But we're trying to see if we can ahead of time predict combinations, essentially just by modeling pathways and trying to model how combinations of drugs will potentially block pathways. This is an example here of a list of drugs that are available to target a pathway that we know well. It's a pathway that's involved in B-cell lymphomas, driving B-cell lymphomas. It's called the BCR pathway. What we've done in the past few years was to build a computational model of a BCR pathway that has essentially a lot of connections, really sort of is trying to mimic the way that the signal is transduced from the top of a pathway to the bottom of the pathway, feeding and fueling cell proliferation. We know that lymphoma cells where this pathway is active are addicted to this pathway, and all these drugs that I showed you are essentially trying to do one thing, which is to block this pathway. And blocking this pathway is well known to essentially be effective in terms of killing lymphoma cells. The problem is that this pathway is very complex, and it's been very hard to block the pathway using single agents. So we've been saying, well, maybe we can model the pathway, try to understand the feedback loops that are driving the pathway, that are essentially present within the pathway, and maybe identify how we can use drug combinations to really shut down the pathway for good and essentially disable these feedback loops that are all over the place in the pathway. To do this, we've been using machine learning and differential equations, machine learning to essentially feed the model to data that we have. We also had to get a lot of data from literature about the different components, parameters of the model. But we ended up with models that are actually pretty effective at recapitulating the known signaling dynamics of a pathway, and therefore that we thought would be effective in terms of predicting how to combine drugs to block the pathway in an effective manner. So we made predictions about combinations of drugs that would be effective in terms of synergizing and blocking the pathway in a very effective way. As a control, we also made predictions about combinations of drugs that would antagonize each other, essentially enable sort of further signaling within the pathway. And essentially, we did experiments in cell lines looking at these combinations that we predicted, and we got pretty good response, pretty good sort of correlation between the predictions and the experiments. Meaning that essentially the combinations that we predicted to be synergistic are actually highly synergistic, in cell lines, and the combinations that we predicted to be antagonistic are actually highly antagonistic also in cell lines. This gives us confidence that we can use this kind of model, to predict how to combine drugs, moving forward, assuming that we can get enough mechanistic understanding of these pathways, which again takes a long time and a lot of work, but potentially is feasible, as you can see here. So just a few more slides to show how we are looking at tumors from different angles. We are completely aware that tumors are complex ecosystems. Tumors consist of lots of tumor cells, but a lot of immune cells and non-immune cells, for example, endothelial cells, which we know very well. So we are really sort of thinking that we need to embrace this complexity, not shy away from the complexity, but really sort of embrace it and model it. So what we're doing is, essentially in a variety of different ways, is to try to capture the different components of the microenvironment of tumors. One of the reasons why we want to do this is because we'd like to be able, for example, to predict, based on the microenvironment and features of the microenvironment, what patients are likely to respond to, for example, immune therapies, which fundamentally are geared towards targeting the microenvironment, targeting T cells that are becoming effective at killing cancer cells. As we know, to predict response to immune therapies, you essentially have to understand the immune system. And as we know, the immune system is very complex, has lots of components, and it's even more complex within the context of tumors, where you have essentially features, parameters on the tumor cells side that are important, but also features important on the immune cell side. So what we're trying to do in tumors more and more is to essentially get an immune landscape analysis of tumors, try to get a sense of what kind of immune system, what kind of immune parameters and features a tumor has. We're doing this in different ways. So essentially you can do this with measurements, new measurements that you don't necessarily get from whole exome sequencing and RNA-seq. So one type of measurements that we make more and more now is direct T-cell receptor sequencing. This is here examples of precision medicine patients where we performed T-cell receptor sequencing. And as you can see here, different patients have completely different types of T-cell clonalities. These measurements here essentially measure the diversity of T cells that you have within a tumor. You can see different patients, completely different types of clonalities and abundance of different T-cell populations. We are also looking to take mutations we see in tumors and predict which ones of these mutations are likely to be neoepitopes. And we find a lot of neoepitopes in most of the tumors. This also is a component that we think is important, and we calculate this for every tumor. We've been also looking at, as I said at the beginning, immune deconvolution, trying to understand what kind of immune cell population you have within the tumor. And we're also working on new algorithms to do this in a very effective way. For example, taking into account the hierarchies of different immune cells that you have in the immune system, sort of taking into account the fact that you have essentially, sort of types of immune cells that are very similar to each other and immune cells that are different from each other. By modeling this hierarchy, you can actually get better deconvolution results from bulk sequencing. So all of these analyses we try now to do on every single tumor. Our goal is to predict what patients are going to respond to immune therapies. This is just here sort of very preliminary results showing how we can integrate those different measurements. Again, neoantigens, immune cell deconvolution, T-cell receptor sequencing. We combine these informations using machine learning. We do this in the context of trials where we have information about who responds, who doesn't respond, and basically we learn how to combine these features so that we can predict responders. Long story short, we don't have a lot of patients to play with in those studies, but in test sets where we essentially did the training on some patients and applied the machine learning to other patients, we actually see a close to 90% accuracy in terms of predicting who's going to respond to checkpoint inhibitors. Again, this is something we are trying now to do on the larger cohort. Just very briefly, we're doing a lot of work, again, to measure what's happening in tumors in a more effective way. From single-cell RNA-seq to CyTOF, we have an imaging CyTOF that we are starting to use now to get a spatial sort of analysis of tumors. And I'll come back, at some point to tell you more about the data. But really, when you look at single-cell RNA-seq tumors, and I'm sure many of you have been exposed to this kind of data, we see a tremendous amount of diversity within tumor cells and populations, but also within the immune sort of component to tumors and the non-immune component of tumors. So there's a lot of work that can be done essentially to understand this complexity, understand, for example, what kind of crosstalk you have between the tumor cells and the non-tumor cells. Likewise, this is something, I'm not going to go into details about this, but in collaboration with others at Cornell, we've been essentially studying these crosstalks between different subpopulations and look to study how we can leverage those crosstalks to better kill tumor cells. I'm just going to conclude, and clearly my time is up. Patient-specific avatars enable essentially these mini clinical trials. I think it's a very powerful paradigm where you can essentially conduct mini clinical trials for one patient at a time, test thousands of molecules, thousands of combinations. I think it's really the future of cancer care is going to include this kind of tool. We have to get better before we can essentially use this in patients. But really, the data and really sort of points at this being a great tool for identifying what patients are going to respond to. Single-cell technologies. I mean, I showed you just a few data points on this, really providing us great insight into the complexities of tumors. And telling us about potential crosstalk between tumor cells, non-tumor cells that we can leverage again, potentially identify new targets, a new way to target cancer cells using this knowledge and information. And again, the key here is really that this is complex. I usually, maybe not in this audience, but I always get a feeling that people don't like when I say that this is complex. Actually, cancer is complex, but that's a reality. Our sort of mandate, our focus, our mission is really to embrace this complexity, model this complexity, and think we have the tools, we can measure this complexity not really well, and I think we should not shy away from it and really embrace it. And obviously, all of this will require very tight collaboration, so integration between experimentation and analysis moving forward. And that's the end of the talk. This program that we have is fundamentally multidisciplinary. It has a clinical component, it has a laboratory component, has a research component, has a computational component, and these are some of the leaders of the program on this slide here. But obviously, lots of people are doing the work. I only have just a few slides about some of the people here, but essentially it's very much a team effort, and lots of people from different fields involved in different components of the work that I showed today. So I'm going to stop here, and I'm happy to take any questions. Thank you.
Maybe just a couple of questions.
So nice talk. So it's not surprising in the organoid you see preferential sensitivity to tremetinib in the KRAS mutant organoids. One of the concerning things is that, of course, the MEK inhibitors have not shown efficacy in clinical trials. So are you able to observe these differences as you move to the vascularized organoids that you're able to capture this?
Yeah, so we need to, I mean, the reason why we want to bring robotics, essentially to be able to do those high throughput screens, essentially in an effective manner, but also, do a lot of different conditions, a lot of different sort of co-culture, situations, so that we can really broadly understand the impact of vasculature. And so the answer is basically that we don't know yet, but I think it's a great question, and I think hopefully we'll know the answer soon, to this question. Yeah.
Can I ask you a question? The zebrafish are really cool.
Yeah.
I really like that.
Yeah.
Can you use the zebrafish for immunotherapy studies? Is that
Right.
the idea?
So unfortunately, sort of not yet, right? So we're actually leveraging the fact that at the early embryonic stage, the zebrafish does not have an adaptive immune system. They have a kind of a, sort of innate immune system, but not an adaptive immune system. And therefore, they're not really able to fight back, and essentially get rid of a tumor. So it's possible that, lots of things are possible, right? I mean, so one possibility would be to co-inject, immune cells, human immune cells, together with, sort of tumor cells. Mm-hmm. And there's a possibility that those immune cells would also be sort of maintained alive, as a result of the survival signals that you have within the zebrafish environment. And by the way, so I didn't really mention this, but I think that's also the hope when it comes to these vascularized, sort of, organoids. Because we also have data showing that you can essentially keep T cells alive if you co-culture them with endothelial cells. So one idea is that the endothelial cell co-culture system would allow us to maintain the tumor cells alive in a kind of physiological environment, but also potentially to maintain immune cells alive and essentially enable reconstitution of an environment that's very similar to what you have in tumors. So I think these tools enable a variety of different opportunities to really get to the point where you can use those tools for IO analysis. Yeah.
Oh, maybe we should introduce ourselves.
This was wonderful to hear something beyond the standard of care. I have a kind of wild question. Are you able to get, I'm thinking of Herceptin and bladder cancer. Mm-hmm. Are you able to get the company trials or some indication of what they found that many years ago didn't work or something? And I suppose the FDA record, whatever, but the company would have given up on it. And in this age, they might be glad for some kind of an extension of use. So is there some way? I mean, have you tried, or have people just hit a stone wall?
Yeah, so it's obviously a complex question. Every time that you talk about industry, I mean, the answer is, often sort of, complex. But we have a whole program that I haven't talked about today about drug repositioning, which is essentially to take drugs that have been shelved for a variety of reasons. Usually it's, failure of clinical trials in phase 1 or phase 2 or phase 3, and see how we can use them in a different context, and potentially reposition those molecules, sort of for a different indication. And so, we have had some success in some drugs. Usually these are drugs that are available sort of, not, necessarily, in a too sort of difficult way, but getting access to the, companies' sort of pipelines or, shelved, molecules is never very easy. And you basically need to have kind of a close relationship with the companies and sort of, get to know them and get to know the people who've developed, the molecules so that we can get access to those compounds. I think it's been done in the past. It's a little difficult, but I think it's feasible. And again, we have the tools now I think we definitely have the tools to reposition molecules using, this organoids, for example, and others. So these are things that we'd like to do a lot more moving forward. We have the tools to do this, but getting access to those compounds can be difficult, yeah.
Is that a very short question? Because we are a little over time.
Sorry.
I like your cultures with endothelial cells because they resemble fibroblasts or myofibroblasts. I was wondering if you check CD31 or if you perform endothelial assay. And the second question, very very short.
Just answer what.
It's supposed to, yeah.
Yeah.
So you're talking about the endothelial cells, but so the endothelial cells, so they're co-cultural endothelial cells. So they are, we have all the markers, that endothelial cells express and so on. They are essentially, they are not from the same patients. They are actually engineered endothelial cells. And so we know that they're endothelial cells without any issues. And essentially, they form these tubes, this sort of, that is also sort of features of endothelial cells. Yeah.
Okay, thank you. Thank you very much.